Right, we've looked at this idea of monoploidy already when we looked at haploid male bees during parthenogenesis in asexual reproduction. Looking at it here in terms of chromosome numbers, we're going to talk about monoploidy. So an individual that contains 1/2 the normal number of chromosomes is monoploid. So it's got one N. Remember, N is how many different chromosomes you've got, and we've got in humans 23 different chromosomes, so that would be our haploid number. So rather than saying that this male drone down here is haploid, which he is because he's got one set of chromosomes, we can also describe him as being monoploid. So he's displaying monoploidy in that he is a viable bee that's got only one copy of all of the different chromosomes. We're just introducing some new language here, things that are similar but different. Monoploids are very rare in nature because if you've only got one set of chromosomes, if there's a lethal variation on there, then it's going to be expressed. So if there's a disease on here, there's no other chromosome to mask it. So if the mutation is there, generally the organism will die. So this haploid animal can also be described as being monoploid, mono meaning one, and it's got one set of chromosomes. So we've talked about this already. Insects produced during parthenogenesis are monoploids. They have one haploid functioning set of chromosomes. So we can think about haploid being a description of how many chromosomes, and an organism with a haploid set of chromosomes we describe as being a monoploid organism. You're a diploid organism. So you're diploid. We have a little look at my design down here. So if you were a haploid, we describe you as a monoploid organism. You'd have one set of chromosomes. You're diploid. You've got two of all of them. But you can also be triploid and tetraploid and hexaploid and so forth. There are organisms that can have more than two copies of each chromosome. This is known as polyploidy. When we have a little look here, we can see that things like bananas and strawberries have got many copies of the chromosome. So a strawberry can have up to 8 copies of the chromosome. So polyploidy is referring to organisms that have got more than 2 sets of chromosomes. Very common in plants, would be lethal in humans. The advantage is that it makes the plants bigger and the fruit bigger, and it's something that's been bred in into plants, can have a negative effect on fertility. What do you need to know? You need to know that organisms can be polyploid. What does that mean? It means instead of being diploid and having 2 sets of chromosomes, which most living things have, they've got 3 or 4 or 5 or 6 copies of the different chromosomes. Polyploidy. Now, aneuploidy. It's going to be a little bit of a head spin, all of this, and you're just going to have to take your time and go through it. Aneuploidy is the presence of an abnormal number of chromosomes in a cell. So we're not talking about having an extra set. We're talking about we're missing a chromosome or we've got an extra one. If non-disjunction occurs in meiosis, so non-disjunction meaning that the chromosomes don't separate properly, if that happens in meiosis, then you could have 45 chromosomes in one cell, 47 in the other, when we would expect to have 46. So this is normal meiosis here. Have a look. We have in meiosis 1, homologous chromosomes separate. In meiosis 2, sister chromatids separate. And we end up with one copy of all of the chromosomes in the daughter cells. Taking you back, yeah? So we should have N. 1 from chromosome 1, 1 from chromosome 2. If we have non-disjunction in meiosis 1, so what does that mean? It means when they lined up at metaphase with their pair, so here's pair number 1 and pair number 2, pair number 1 went this way and this way, but by mistake, the two number 2s went to the same end of the cell. So now instead of there being 2 chromosomes like there are here, there's 3 in one and only 1 in the other cell. That's aneuploidy. There's been a mistake at meiosis 1, and as a result, this one's got an extra chromosome instead of the 2 that it should have. It's got 2 copies of chromosome 2, and this one is missing a copy of chromosome 2. That's if it was a fault at meiosis 1. If it was a fault at meiosis 2, so in meiosis 1, homologs lined up in the center, anaphase 1, the homologs got pulled to either end of the cell. Meiosis 2, the chromosomes line up across the center. Sister chromatids separate to either end, and in this one, sister chromatids didn't separate. Okay, so this is what should have happened. So we've got the 2, this happened properly. Here's the 2 cells that are now going to go into meiosis 2. This one, these sister chromatids have separated to either end, and we have 2 chromosomes like we should in the resulting gametes. Here we've got this pair of sister chromatids didn't separate, so we've got 3 chromosomes in here. Chromosome 1 and 2 of chromosome 2, and in here only 1 chromosome 1. This is called aneuploidy. This not separating properly is called non-disjunction, and I'll add all of these into the Quizlet, which might be a good thing to be doing to learn the language. All right, that's just a bigger one that I should have gone through that one with you, but you can go through and tell yourself the story. Here's the story of 2 pairs of chromosomes that lined up in the middle in metaphase, and in anaphase they got pulled to either end of the cell. Here's the story of 2 chromosomes that lined up in the middle. In anaphase, this pair got separated, but these 2 went to the wrong end of the cell, and you ended up with an extra chromosome here and missing one here. Here's the story of 2 chromosomes that went into meiosis 1, and the homologs separated. They went into meiosis 2, and in this cell, the sister chromatids separated like they should, but in this cell, these sister chromatids stayed together, and we ended up with an extra chromosome down here. You need to take your time to work your way through that and make sure you understand it. Draw it if you have to. It's really important that you understand it. Just a little summary of the different types of ploidy. So up here, if this is a diploid organism, we can also have polyploid organisms and tetraploid organisms. So this is where they have, as they should, different numbers of chromosomes in their cells. This is an aneuploidy arrangement, showing you where, instead of being diploid as it should be, some of them have got the wrong numbers in their cells. So this one here hasn't separated properly, and it's got trisomy. It's got an extra chromosome here. All right, so chromosome 1 separated properly, chromosome 2 did not. We've got 3 copies of it. Chromosome 3 separated properly, chromosome 4 separated properly. So this is trisomy because it's got an extra one. In this one, chromosome 1 didn't separate properly. We've only got one of them. We've got monosomy chromosome 1. The others are in pairs as they should be. So this side's looking at aneuploidy, and this side's looking at polyploidy. Okay, I'm not going to read all of this to you, but we've got just a summary here. Now, there are some diseases that are associated with having the wrong set of chromosomes. If you've got trisomy 21 as a human, you've got an extra chromosome 21, then you have the disease Down syndrome. So non-disjunction of chromosome 21, where we end up with usually the egg that had an extra chromosome in it, the sperm joins, and we end up with 3 copies of chromosome 21. The baby will develop, but it will have all of these genetic defects. It will have a faulty heart, it'll have issues with its fingerprints, mental retardation, flattened forehead, a whole lot of different features. Viable, child will survive, but it will have all sorts of deficits in the way that it develops. So trisomy 21, tri is 3, we've got 3 instead of 2 chromosomes there. It's not a diploid, it's triploid. Klinefelter syndrome happens if you've got a problem with your sex chromosomes. So 3 sex chromosomes for Klinefelter syndrome. So we've got 2 X's and a Y. So they've got the 2 X chromosomes that give you the female, and then they've got the Y that gives you the male. So they appear to be male. They have small testes, they have a penis, They have fat distribution similar to a female body, thicker across the hips. They have some breast development, reduced facial hair, reduced body hair. And typically they're very tall. Don't know why I do that. Turner syndrome, you can watch the video. I'm not going to watch that with you. But Turner syndrome results from having only one X chromosome. So girls who've got only one X chromosome, short stature, non-functioning ovaries, infertility, puffiness around the neck, and so forth. You can watch the video there for yourself. Alrighty, so this is known as Turner syndrome. Another video on it, and here's what it looks like. So this is a monosomy of the sex chromosomes. There should be two X's there and there's only one. There's enough genetic, so she's got enough chromosomes to make her be a functioning human. She's just missing some of the genes that she needs to become fully developed as a female.